Yeah.
What did they tell you about the mics? What are you going to do for mics?
I'm going to do that.
Are you going to wander, or are you going to...
I wasn't going to wander, but with the questions, I'll still try to do that, the stand mic.
Okay. No, I'm considerably shorter than you. Let's see how this is going to go with you. Good evening, everybody. Welcome so much to our Fall Badger Café. I think I caught most of you on the way in, and so many of you had been to one of these before, which is exciting. We've been doing these since 2008, believe it or not. My name is Erin Elliott Kazik. I'm an 03 grad, and we started these in 08, and it was just kind of an idea. Like, you know what, I think that might be, we love doing game watch parties. We love all the other activities that we do with the chapter, especially our social aspects, raising money for scholarships for Brown County students. But we thought, let's try something, in the education bent a little bit. And we planned an event at Not by Bread Alone. So those of you who are familiar with the area, especially as Robin and I may know it. And it's not a big place, and we just figured we'd probably get about 20 people. And people kept pouring in and pouring in and pouring in, and pretty soon we thought, oh my gosh, I don't know how I'm going to get to the bathroom. So we thought, there's something here. And ever since, we've been having them in spaces like the Brown County Library's auditorium, which is large, and obviously this beautiful event space. So thank you so much for coming out tonight. We will continue to do these once a semester, so please keep watch on your email for further information about these as time goes on. We're very lucky tonight to have Professor Fletcher, Jason Fletcher here from the University of Wisconsin-Madison. Most of you in the room are alums, so woohoo! We're so excited that you are also an alum. Sometimes when I do these bios, I look down and I see where the people are from and where they went to school, and they're not Badgers, but we don't have to worry about that tonight. So let me give you a little bit of his bio. As a professor at the La Follette School of Public Affairs in the Department of Sociology at UW, Jason is researching questions about how public policy intersects with genetic data, what our genes can predict about how society functions, and how we should use these data responsibly. He specializes in health economics, the economics of education, and the child and adolescent health policy. And his research includes examining social network effects on adolescent education and health outcomes. He earned a BS in Economics and Public Administration from the University of Tennessee, Knoxville, which I will read past very quickly, and his MS and PhD from the UW-Madison in Applied Economics. So his talk tonight, which you all know, because you all obviously came here because you're fascinated about this presentation, Molecular Me, Exploring the Social Implications of the Genomics Revolution. He has said that it's totally fine to ask questions during the presentation, so we have a curious bunch. We always do. If you have questions, feel free to raise your hand. He will be sticking around for a few minutes afterward, but I have to emphasize to you, because remember, he's going back to Madison tonight. And if you are interested in learning more, he has a phenomenal book, The Genome Factor, that he's actually brought a copy to donate to our local public library. So I will be bringing it over there, and if you would like to check it out, my guess is in the next week or so, you may be able to check this out from the library. So we appreciate you doing that. Without further ado, please let me introduce Professor Jason Fletcher.
Thanks, Erin, and thanks you all for coming. I'm really excited to share some of the research I've been doing. I'll say a little bit about myself first. I'm from a very small town in East Tennessee. I went to UT, like Erin said, and then I came up to get a master's and PhD at UW-Madison, and loved it. Loved the city, loved the state, and had to go to another job for a while. So I was at Yale for 7 years in the School of Public Health, and that's part of how I learned about genetics. I'm a health economist by training. I was in the School of Medicine and Public Health there, and there were probably 3 economists and 1,000, MDs and clinicians and geneticists. So I was kind of overwhelmed by that. And since then, I now have my dream job at La Follette School of Public Affairs, where I can integrate public policy, economics, sociology, and some genetics. And here, I'm just really excited to be a part of the Wisconsin idea, which is to share some of the research across the whole state. So thank you very much for coming. I am used to being both interrupted and have questions throughout, so I'd encourage that. I only just try to get my attention. That's the only thing. It's not that I want to entertain as many questions as possible for clarification or for extensions. I'm super comfortable with that. So thanks again for coming and for Aaron coordinating this. So with that said, so there is a book that I would say that the book lays the foundation for what I'm then going to talk about today. So it's much more both for an academic and non-academic audience. I work together with Dalton Conley, who's a sociologist now at Princeton, and he's one of the few people who, so he got his PhD in sociology in '96, and then he went back and got a second PhD in biology more recently while he was a dean at NYU. So he's a fascinating person. I'll set that aside, and you have to see me instead tonight. So like Aaron said, there'll be a copy of the book shortly here locally, and it's of course available online and so on. So since you haven't read it, I'll summarize it here, which is to say there's about 100,000 words in the book, and if you sort the words by frequency, you can get something like this, which is a wordle. And big words mean that word was used a lot in the book, and smaller words less so. So I take this as maybe showing the intersection between genetics and social science here, where you get a lot of genetic words, but also population and social environment and so on. And it's also a little bit of a diagnostic. When I look on here, I see that we used IE like way too much in the book. So that's like a post-diagnostic to say like you should back off on using IE if that's a big part of your book. But now that we've got that out of the way, you've read the book. I'm going to then make an argument that we're in the middle of a revolution. So that's a strong claim. So I'm going to make a little bit of a pitch on that. So in particular, it's the revolution is emerging from genomics. So I'm going to describe that, but it has a lot of social implications. So that's what I'm interested in. I'm not a geneticist by training, so I'm going to give you a few slides on the basic genetics, and then we can start thinking about what this revolution on the genomic side is going to do to society. So I think that's the point of the book. So within the field of molecular biology, genomics is the study of genomes. And genomes is just a complete set of all genetic material within an organism. So let me give you some facts about what I think is of as the revolution. So let me first remind you of some of your biology or genetic background, which is I'm going to be discussing humans, and so let's reorient ourselves about the complexity or lack of complexity in the human genome. So it's about 3 billion letters long. So in each of our cells, if you dive into the cell, you'll find the nucleus and where all the chromosomes are hanging out. And if you look in each of the chromosomes and maybe line them all up, then you would have the 3 billion or so letters long of our genome. And that's in each of our cells. We have trillions of cells in our body. It's essentially the same in every one of our cells. Some facts, about 1.5 billion miles of DNA in your body. That's 3,000 round trips to the moon. If each of these letters, these nucleotides, was a letter in a textbook page, you could stack the pages 65 feet tall in each of our cells, right? There's a lot of information here. And so another reminder is that although that's a lot of information, 99.5 or so % of all the letters, all those 3 billion letters, are identical across everyone in the room and everyone in the world. So there's an immense common humanity when you think about the letters being so identical across so much of the genome. Now, then I'll throw a little bit of cold water on that to say 99.5% or so, all the letters are the same across all humans. 98% of all our letters are the same with chimpanzees. So there are differences, like the letters have meaning. So the letters can matter. And in particular, there are instances where small variations in the human genome really matter. So some of the letters can matter. And I'll show a couple cases where a single letter really can change your life. So sickle cell mutation is an example. So these are letters. All your letters are either A's or T's or C's or G's. And we have a normal cell letters, and you see all C's and A's and T's and G's. And here are the proteins that those are coding. And you can see a single letter, T to A, across two humans has this massive impact on the way your cells look. And here's a sickle cell. And so although we have 3 billion letters, most of them are the same. Some of them really matter. So that's one instance. Another instance is there's just one letter that determines whether you can digest milk after weaning, whether you're lactose tolerant or not. So this is just a letter. And many thousands of years ago, no humans had that letter. And then there was a mutation that swept Europe and Asia separately, that now a lot of humans can digest milk post-weaning. So those are some instances where a single letter really matters. And the other thing what I'm going to be describing today are these letters, and a reminder that the letters are permanent. These are letters that you had when you were a fetus, when you were an embryo, and you're going to have these same letters basically your whole life. Okay? So I have an asterisk there because at the end I might talk a little bit about gene editing. But that's in the near future or approximately. But what I'm going to be describing is something that's a little bit more permanent across many generations. But the letters you should think of as soon as you were an embryo, you had the same letters that you have now in all your cells. So they're permanent in that sense. So I haven't told you why this is a revolution yet. I've basically given you some facts that were true 100 years ago, 20 years ago, and 1 year ago. Why do I think there's a revolution? And why do I think the revolution has tremendous social implications? So that's why I'm going to set up in a few slides the movement of DNA to social genomics. So this is my claims for the first few minutes of the talk, which is I've given you some genetic facts. Those haven't changed in a long time. Now I'm going to tell you what's changed and some of the implications for what's changed. And the first one is going to be technology. Has just improved at such a scope and scale that we don't even know. It's hard to even describe it. So I'm going to describe it in a few slides of how fast and large the technological improvements are. Then I'm going to have a few slides on statistical improvements that take the technological improvements and make predictions that we could never make before, but that we can make now. And if we add those three places up, then you've seen the genetic facts. I'm going to give you some facts on technology and statistical improvements. I'm going to then outline some of the many new policies and this is moving from genomics revolution to social genomics revolution. So what are the technological improvements? I'm going to focus on a couple. One is about price and cost. So this slide is not about genomics. This is just reminding you that prices change over time. And so some things get more expensive. So college tuition, if we go back 20 years versus now, college tuitions have increased by 150%. That's a lot. TVs have, the prices of TVs have been reduced over 20 years by a lot. But this is normal change when I compare it to the changes that have happened in genomics. The cost per genome over the last 15 years. So Moore's law is maybe the fastest increase in technology that, and this is about computers. So every 18 months or 2 years, processing speeds. This is something that was observed about 40 years ago, so it's a little bit dated. But Moore's law is the observation that every 18 months or 2 years, processing power per cost doubles. Okay, so that's really fast every 18 months if you get a doubling. That's Moore's law. The green line is the cost per analyzing genomes. So this is the price of analyzing a human genome has fallen by 100 million times in 15 years. So this is the fastest thing that we know. This is the cost per genome. So this is restating that 15 years ago it cost $3 billion to sequence the first human genome. Now you can sequence your genome for under $1,000 right now. So that's going to have some implications. That size and scope and scale of the costs, which is that it's now super easy to get your genome sequenced and really inexpensive. So these are a couple of the top companies in the United States. And I've just plotted a couple data points where, so 23andMe is one of the companies that if I just go back all the way back to 2015, they had about 800,000 subscribers, meaning that people had contributed biological samples and had them analyzed by 23andMe. Now it's 2 million in 2017. Ancestry DNA, you have those other two data points. The beginning of the year, they had 3 million people who contributed data, genomes. 3 months later, they had another million people who are subscribers. And then we have an upstart family tree who have almost 1 million by August. So just those three data sets are 1 in 50 of the US population. And these are all in private hands. I have a quiz, a one-slide quiz. What's the single largest DNA set, largest data set of human DNA? Yeah?
The Human Genome Project?
It's a good guess. Any other guesses? So it has to be bigger than these. Like the number of people in them have to be bigger than this to be larger. Yeah?
Military?
That's close to. Anything else?
The VA?
Good one. These are all not right, but yes. So you're actually getting close. So who has ever seen a program called CSI? What are the words that you hear every CSI episode? CODIS, NDIS, right? Those now, when you add all that information, you have 16 million people in one data set, or 1 in 20 Americans. So that's a big data set. The UK has a relatively new initiative that already has almost 5 million people in it, which is that once you get arrested, even if you're not charged, you get to contribute DNA to a centralized data set. I'll come back to some of the criminal justice issues that this is raising. But so these companies are big, and they're going to be this big. In fact, the projections are, let me unpack this projection, which is we're here in a little bit after 2000, right here, where this line starts to diverge. So right now we have that many million genome sequence. The projections, either conservative or if the line just continues up, by 10 years from now, is that we'd have somewhere between 100 million DNA samples to 2 billion. Like that's the range in 10 years. 2 billion is a lot of people. So, but even 100 million is a lot of data, and so this is opening up questions about what are we going to be using this data for. So that's part 2, which is statistical analysis of these data. So the part 1, part 3 is statistical improvements. Part 1, genetic facts, is to remind us. Part 2, technology improvements, which I just described, huge reductions in costs, huge increases in the availability or the demand for depositing your DNA in these massive data sets. Now I'm going to walk you through a couple slides of what companies and researchers are doing with these data sets that didn't exist at all just a few years ago. And so that's about predictive analysis. So there's both going to be a population level component here and a personal level of predictive capacity. So this slide is just one. You don't need to read the fine print. What I'm describing is just 2 months ago, the UK released a single data set of close to 500,000 people with that many outcomes. That's everything from height to body weight to IQ to years of schooling to are you married, how many kids you have. So several hundred of those traits. And each person, think about how big this data set is, each person has 30 million pieces of the genetic data analyzed, to analyze. So you have almost 500,000 people multiplied by how many traits they have for each person, multiplied by how many genetic variants they have for each person. So this is one huge data set that's now, if I give this talk a year from now, I would say more about it. This was just released 2 months ago, and all of us, you're already going to see some of the implications for analyzing these huge data sets of many, many traits. And this is just one data set. Other countries, China has emerging data just like this, of this scale, of just hundreds and thousands or millions of people, all with a lot of genetic data, all for the purposes of prediction of an outcome. And in this case, it's mostly health outcomes. So what do you do? I'm going to just give a slide or two on how findings or how discoveries are made in genetics these days, which is you take a data set like that, which is huge, and you just scan the whole thing, chromosome by chromosome. This is a picture of all your chromosomes. Each chromosome has many, many letters on it, and you go letter by letter and ask whether the letter A versus the letter T, or the letter C versus the letter G at each of these locations predict X, where X is heart disease or any health trait, height, years of schooling, anything. And you do this data mining for novelty. You can be doing 10,000 or millions of statistical analyses, and then you have a lot of results, and you have to comb through, like, what did I find? So I'm going to give you a slide of how that's presented, which is that you have a big data set, you run 10,000, 100,000, a million statistical analyses, so you have a million results, and then you're going to plot it on what's called a Manhattan plot here, which is that each of those points is its own statistical analysis, asking whether an A versus a T seem to increase the risk for schizophrenia or body weight or whatever. And you're going to plot that like this. So these are hundreds of thousands of individual statistical analyses plotted, and what you're going to do is going to look above this line for statistical significant findings and find some, like this one, and then go ask whether that finding is true in other samples. So it's an out-of-sample prediction. So let me give you a flavor of what this looks in practice. So sometimes you find something really clear, which is you scan the whole genome, and there's this one massive tower in this Manhattan plot. And in this case, it's a plot showing the genetic architecture of whether you smell, when you smell urine of someone who's just eaten asparagus, if you notice that. So that is a genetic trait, and there's a very precise area in your genome on chromosome 1 that lights up. So people who have an A or a T at those locations smell urine differently for people who've just had asparagus. Okay, so sometimes it's like super clear of where in the genome something matters. Okay, this next figure is just released 2 days ago, which is food allergy. So there's both a little bit more. So let me go back. There's nothing anywhere else. Here, there's mostly something right here on chromosome 6, but you see some scattering around here. So it's both a very strong one signal for food allergy, and but also some potential for complexity across the genome. There's other places that might be hits, and you see something like peanut allergy with a peak here that's definitely there, and then some other peaks that might also be true. Egg allergy, you see the single peak, but also some background peaks. And then cow's milk allergies. So this was published like a couple days ago. But so you can see these. In fact, I think I'm going to show you a slide. There's something like 13,000 of these papers. It's a lot of papers because every outcome you do the same type of analysis and you plot it in the same way to see where there might be something in the genome to then follow up later to say, well, why is that true? Why is that particular location seem to be heavily related to something like food allergy? What's the biology behind it? So it's not just health, and now I'm going to be shifting closer to social science, because these are straightforward health science questions or of interest. Food allergies, the asparagus smell in urine, that's not quite a social science question. But there are social science questions here. So this was a full year ago, this picture was presented, which is this is how far in school you go. This is your educational attainment, whether you go to college or not. And what you see here are a whole lot of peaks all the way across the genome. So this is a so-called complex trait, which means that there's many, many hits across the whole genome. And you think about educational attainment, how far it is maybe away from your letters, like what all these letters are doing across your whole life course that then leads you to, or is related to you going to school longer versus shorter. This was done in a set of samples that in combination was almost 500,000 people, and the headline was 74 independent locations in the genome predict how far in school you go. So now we're closer to social science, a combination of genetics and a trait like, or a choice/trait like educational attainment, and asking some of the genetic bases of this. So when it is complex like this, where it's not just a single peak, then what social scientists and geneticists do is to combine all the information across the whole Manhattan plot into a single number. And that was there a question? I should stop. Let's just take a pause and ask if there is a question. Okay. So you summarize all this information. You have all these A's versus T's all across your genome, all across your chromosomes, hundreds of thousands or millions of potential signals here, and you sum it all up in what's called a polygenic score. And I'm going to describe that in a little bit more detail now, or almost now. I'm going to just say that this was a whole year ago. This was May 2016. So between in the interim, there's been a whole lot of other findings that are related to this. So there's one on IQ, for example, that was in Scientific America where there were 22 genes associated with IQ. So it was very much a Manhattan plot, but instead of educational attainment as the outcome of interest, it was IQ tests. So again, we're very much into social science areas. So these genome-wide association studies, or GWAS, have been conducted now for about 10 years. And so this is a plot of the accumulated knowledge over the last 10 years. So 3,000 different genome-wide association tests over these 10 years. And there are over 39,000 different locations plotted towards food allergy, to the asparagus finding, to years of schooling. When you add them all up, that's a lot of findings to work with. And so how people work with it now is to summarize it into a single index. So it's like your credit score. You take, but instead of your credit score being all your past purchases and whether you pay your account on time, it's all your letters. And you sum up all the letters and all the risk across your genome into a single score that looks like this here. So it's in fact normally distributed in the population, something like education. And then here is what's done with the score is, everyone in the sample, you can score them, and then you can ask whether you have a high score or a low score, and then make medical decisions based on that scoring. So this on the lower left is what's done. You might have a high score versus low score. That puts you at higher, low risk for a certain cardiovascular illness, say. But I'm not going to be talking about cardiovascular. I'm going to be talking about social science and education now. So in fact, what you can do is that when you send your DNA to 23andMe to scan, they send back the results to you. So you get a file, or you can ask for the file. It's not automatic. And you have all your letters, and you can create your own polygenic scores. So, for example, there's a site by a group at Columbia University and NYU where you upload your data to DNA Land, and they spit out the lower right-hand side, which is, this is your predicted years of schooling. So we're very much personalizing genetic information now. So this is a friend of mine who who has a PhD, so this is wrong. But nonetheless, you can do this for years of schooling, for IQ, and for many other traits, personalized, right? So this is your letters, you upload it to this website, and this is not a company. They do this for free. It's researchers at Columbia and NYU that are amassing their own data set. You have here, this is a couple months ago, that they have about 60,000 people who've uploaded their information, and they send, and the reason people would want to do that is to get this type of prediction back. So that's one thing people are doing right now with, yeah?
Are there predictions statistically high? Are they on the head with what they're predicting?
I think you're asking about accuracy? Yeah. So I think it depends on what you what you expect. So this is a prediction for you only based on your A's and C's and T's and G's, nothing else. Okay? So what I'm going to describe as I go forward is how much of the variation in the population can this type of work explain in years of schooling. So I know that the answer to that is about 12%. In the population, a person who can do this analysis, if they only have polygenic scores, this is the summation of all the letters into a single credit score, that one variable that you created only based on A's and C's and T's and G's in the population can predict about 12% of years of schooling. So it's almost the single best predictor of your years of schooling. What's your best predictor? Your mom's years of schooling, yeah. So parents. But it's really close, actually. So mom's and dad's years of schooling predicts about 15% of the population variation. A's and C's and T's and G's only predict about 12% right now. And it's going to beat mom's years of schooling soon. So as the single one variable predictor of years of schooling, this is going to be the winner like in 3 months. Yeah, question?
When we're talking about these companies that are analyzing, or the companies that are doing the testing, what percent of the exome are they looking at? They're not looking at all coding DNA, are they?
It depends on the company. So what they give you back is a SNP chip. So what they're doing is looking at so think about that big stretch of A's and C's and T's and G's. And recall that almost all of those are the same between you and me. So those are not the letters that they want to look at. Right? They want to look at places in the genome that actually vary across the population. So the actual number of letters that they'll analyze are in the hundreds of thousands or millions.
Are any of the labs doing sequencing like that, or are a lot of them doing targeted genotyping?
So when you say labs, do you mean the companies?
Yeah, sorry, the companies. I'm in genetics, so
Yep, no, I'm getting that.
I get a lot of questions from people, so maybe you can teach me what to say.
So the companies right now charge $100 or $200 to do this. So they can't do whole genome sequencing, which is closer to $1,000, $800 maybe. So it's close where they would charge. So what they're doing, the companies are doing right now is, you contribute your saliva in a cup, and they can hold that, and they do. And right now they're using it on a letter chip, a SNP chip of hundreds of thousands of variants, and that's what they give you back as data. Eventually, they'll want to go in and sequence it, especially, and so these companies do special collections for particular diseases, for example, and so on. So does that answer your question? So it's a SNP chip of hundreds of thousands of letters, and as the cost of sequencing goes down, sequencing means every letter, then they'll do that.
Basically, there's still a lot to improve upon.
Depends what you mean by improve. There's more information to gather, definitely, and that's where things are going. It's partly waiting on the costs to continue to fall, and it's a little bit of a cost benefit I'm an economist of if it costs the company or you $1,000, what's the added benefit of getting all those extra letters for a typical person versus what's the cost is $1,000? What's the benefit to my study? So maybe they would sequence people who have certain diseases very closely, and people who don't, they would just give you the hundreds of thousands of A's and C's and T's and G's. Other questions right now? So what does this allow, right? What does a picture like the lower right allow? It allows, in part, for good and for bad, a shift in how we can discriminate across people. Most of this discrimination in society, you might think now, is group-based. It's women or men, old versus young, white versus non-white, within Hispanic. Whatever, pick your favorite discrimination. It's often group-based. Now it can be individual-based. Now it can be personalized. Because this prediction is for you, it's not for people like you, it's for you. So I'm going to give you some examples to start thinking over, and because these are very much in the ongoing policy domain. The first one is probably obvious to you. What are we going to do about personalized discrimination in terms of health insurance and employment? Right now, the answer is we have a law against discrimination based on genetics, which is GINA, which was signed in 2008 by George W. Bush. Just a few months ago, that law was going to be overturned by one of the repeal and replace efforts. So it's very much a frail or fragile law, because once it's gone, in that law was the ability to employers to not force you to give your DNA, but compel you because you can eat. So if you give your DNA to your employer, you get a different health insurance rate, or if you don't. So this is very much something we should think about how robust we want our laws to not allow this type of personalized discrimination. So that's one. There's going to be many issues. That's one. The other one is how is this going to shape the dating and marriage market, the personalization of information for your Alzheimer's risk, as an example. So I have an OkCupid, which is an online dating site that has a lot of information about this person. I just showed you that you could also upload the lower right prediction. You could do that for future income, Alzheimer's risk, longevity, IQ, fill in the blank. There's hundreds and hundreds of polygenic scores, these summary scores that are predictive about you now and in the future, that nothing stopping that type of information from going on these type of dating sites. And in particular, you might even think that companies in the future would use that for better matches. They can right now, you might fill out a survey about the types of partner you would like, but a company that had the DNA and the survey could merge them and say, who are you compatible with from the DNA perspective as far as your future children are concerned, and from the survey perspective. The technology is now there. The question is, what's going to stop this from happening exactly? So that's dating and marriage. What else might we predict? So I'll give you a hint, and this is going back to the first couple slides. Remind you, these are the same letters you had as an embryo, right? So what else might we want to predict about the future? What do you want to know about your baby? Those are the same letters. You can predict this education score for an embryo. So you clearly can predict some things very easily. Eye color is a genetic condition. Hair color, very genetic. Height, 80+%. These are predictions you can make again as an embryo. Weight, yep. Why not? Yep. What about what they're going to look like when they're 30? Is there demand for this? In looking up this, I found something funny from about 70 years ago, which is a long history of the desire for parents to predict their children's characteristics. What I'm showing here is Dr. Seuss, who had a patent for this device. And what the device did was took a picture of each of the couple and tried to predict the face of the child. Now, the technology back then was different, which is I take two pictures and I push them together, and then a lot of the children, the predictions of the children's faces have mustaches, even if they're girls, right? So it was, it was a mixed technology back then. It didn't take off. He in fact didn't pursue this patent much further than 1939 World's Fair. But it tells you, the deep demand maybe for some of this technology. How far in the future is this technology? This is from one of the top scientific journals, and it was published June 26th of this year. And this is just based on A's and C's and C's and G's, predicting your face from your DNA versus your actual face. So you have that on the upper versus the lower. And again, these are the same letters you have when you're an embryo. So that's what I mean by you want to predict the face of your future child. So what's going to stop that? That's a question. It's not because we can't do it. We have a technology for those who use IVF, in vitro fertilization, where you have embryos out of the body and you can genetically analyze them. It's called pre-implantation genetic diagnosis, where just a few days after implantation, you can sequence the DNA of that embryo and do everything I just described. Right now, we just use it for certain genetic conditions, typically, but that's just our current choice. There's no technological reason that you can't do all this when selecting the embryo for implantation. So there's a lot of policy here to think about. And it's not that this is a policy to think about, in 10 years we can get our act together and figure out what to do. It's right now. People are using this not only in the United States, but also around the world, especially in China, to determine the future children. So this is an example that of two parents who had a high risk for cystic fibrosis. used IVF to screen out the embryos that were going to have cystic fibrosis. Now, seems perfectly reasonable thing to do, but the same operation or the same analysis can be done not just to screen out genetic conditions, but any of the phenotypes or the outcomes I was just describing. Certainly sex, eye color, height, and so on. And this is revolutionary. This story was describing the cost to actually do the procedure was around $60,000. The lifetime costs of cystic fibrosis is in the millions. So you think about just the scale of removing genetic disease from the population. So the future is now, but especially not in the United States. The other countries just don't have the history that we have, and you won't be surprised that Germany does not do this at all. Their history, is the extreme of recent eugenics, but the United States has quite a history of its own of eugenics from just 100 years ago or less. But other countries don't. Other countries have a different view of some of this past and don't have some of the supports for people with genetic conditions. So a place like China is on this path of just immense growth of using IVF to screen out conditions, but nothing stops them. And they're very researchers and individuals and institutes in China in particular are very interested in screening in high IQ embryos. So there was just this article was recently published, and it was describing two clinics in China that carried out 40,000 of these procedures last year. And it also noted an anecdote in the United States that further complicates the story, where there was two parents who were deaf who were screening in deafness in the next generation to keep the deaf culture alive, because there are some conditions that could be screened out, but there's some complications of whether you'd want to screen out something like deafness. So the future is here, but even in the intermediate term or in the next few years, there's going to be much large increases in predictive capacity, meaning that the estimates that these polygenic scores are going to have are going to be much more predictive, and they're going to be greater in scope. The types of conditions, outcomes, phenotypes that are predicted will grow and grow and grow. That FACE paper I showed you was just from a couple months ago. Who knows what else is coming out just in the next few months. So it's going to give you a lot of large increase in personal data that you can share or not. That's going to have a lot of discrimination, quote unquote, opportunities. Because I'm going to describe both the positive and the negative of this type of discrimination in just a minute. And I haven't really described at all the issue of privacy here. So that's another huge public policy question about who owns this DNA data. When you send it to 23andMe, they own it. They can do whatever they want. Other companies will pay you for contributing on an ongoing basis. When they use your genetic data, they'll pay you. Not very much. But you partly own some of that money own some of your DNA going forward. And again, once you contribute it forever. These are letters that you had as an embryo that you're going to have when you die, and they haven't changed. So once you make that one choice, it's a permanent choice. So this last bullet point is, on one hand, I think there's a lot of excitement about what's called precision medicine, using genetic data to target treatments. So some treatments, if you get them and you have the wrong letter, it will kill you, right? So you definitely want to be precise there about not giving treatments to people who have the quote-unquote wrong genotype for it. But on the other hand, genotypes might be very valuable in determining what is the frontline depression therapy for somebody. There's many things that many prescriptions one could have for depression. They all have different molecules. Some of them are going to work better for some people than others. That might be a very valuable thing to know. So that's precision medicine. And I think there's, in general, a lot of positive thoughts about that direction. It's certainly going full steam ahead. But what about precision policy? How might we use this genetic information to target other types of non-medical interventions? of schooling interventions, for example, interventions around dyslexia, interventions around conditions that can be highly predictive very early on before verbal communication, autism. What might we do about using this information about targeting educational and other resources? So that's the very near future. Many of you saw that I had a short op-ed in the Green Bay paper about this because we're right here today on the 20th anniversary. I really would encourage you to watch this. I had my students watch this last week in preparation for the 20th anniversary of just how prescient it was. This was before the Human Genome Project was even done 20 years ago. So it wasn't a long time ago. And these are people that you might even recognize. This is Ethan Hawke and Uma Thurman and Jude Law. They're still around. But some of the features of this story that I highlighted in the piece were that, in this future, job applications were just, you didn't have an interview anymore. You gave them a biological sample, and the biological sample was used to predict what type of, what were the good jobs for you and what should you not be doing. The question is really like, what's stopping this now? Children on the movie, children are selected based on the prediction of genotypes. So there's very much like an IVF idea of selecting the embryos that are the ones you want. There's a scene in this movie that I again direct you to where Uma Thurman, steals one of Ethan Hawke's hair follicles, goes to like a 1-hour photo booth, gets the DNA analyzed. He gets a score, 9.2 or 9.3. Nice catch is the lab technician saying. So this is very much prescient movie. Then what are we going to do with that? We have so point 3 are polygenic scores. Point 1 is like, what are we going to do about employers and health insurance companies using DNA to discriminate? Point 2 is the pre-implantation genetic diagnosis that I was describing. And I'm only going to touch on the next slide, but it's it's another huge question, which is gene editing. And I only touch on it now because, on one hand, it's going to change the world, but in a lot of ways we don't need it when we select embryos in the way I was describing. We don't need to edit anything. We could just not implant embryos that have the conditions we don't want. So, like, this is a little bit more invasive way of actually changing the human genome. And then the question here that people are excited about are, what about enhancements to human capabilities? What about switching letters that humans have never had before? Like, all humans are 99% the same, so there are a lot of places on the genomes that we're all the same. What if we switch some of those around? So this was written up recently in August, describing exactly this. Like you see, we're nowhere close to the limits of athletic performance. You could write another one about the limits of IQ, where it's positively selecting embryos based on these predictive scores. This is, there's Bolt, Shaq are these people who look very different than most other people and have very special capabilities. There's partly a genetic basis for that. So this author is very excited about the promise of using genetic engineering for bringing, like he says, new Bolts and new Shaqs. So that's a lot. I have a couple more policy implications that I want to have in your mind before I open it up more fully. And I think of this, as I was thinking about how to frame it, I think of this as like taking just kind of regular policies that we are interested in and really supersizing them in some way. So one, privacy, recall, data breach, 140 million people, right? So this is going to play out over a long period of time. I'm sure it's going to devastate a lot of people. Release, as many of you know, social security numbers, driver's license, addresses. A question is, what's going to happen when there's 143 million people's DNA hacked and distributed? The thing about social security numbers and driver's license and addresses are they're not permanent. Social security number, maybe. More importantly, they're not predictive. Knowing your social security number, like if I knew yours, it wouldn't tell me much about you. Knowing your DNA tells me something about you. It tells me something that's permanent. So what are we going to do when there's the data breach of millions or hundreds of millions of people? Because it's not something you can take back. What are we going to do about health coverage and pre-existing conditions? In particular, as we move to future health conditions that are predicted but haven't yet arrived. Right now, a lot of the discussion is about pre-existing conditions, meaning they've expressed themselves. You have had a heart attack or you've had a birth, a pre-existing condition. What about future health conditions? What about your risk of Alzheimer's being predicted when you're an embryo and how you might be excluded from health insurance your whole life? This is an example. What are we going to do to have that not happen? I think that this is maybe a broader discussion. There's an interest certainly in social mobility, being able to climb the ladder, do better than your parents. How will this discussion change as we shift to predicting these success markers from genetics? It's always been true that genetics has lurked. Genetics has always been a partial explanation of why parents and children have similar outcomes. We're going to really be able to dig into this more as we actually can measure genetics from children and parents to see how this works and how it works with the environment. So that's a broader, not necessarily policy, but a broader question about what we're going to do with that information. So a little bit of restating. We have access. Who will have access to these technologies? I've treated them so far as everyone can do this. But that's not going to be true. Not everyone's going to be able to afford these different technologies. Not everyone's going to be able to screen their children or their partners or themselves. And there's a possibility, because just as an example, the Catholic Church is very much against a lot of these technologies. You could imagine a world where individuals of the Catholic faith do not use these technologies at all, or to a lesser extent, and the genetic conditions that are now across the whole population become more and more concentrated on the poor and on those of certain faiths. Again, you just think about what we're going to do with that information going forward. Who's going to own your DNA information? There was a time where companies could patent your DNA, right? Or that's been overturned, but it's fragile. Maybe it goes the other way. Who knows? What are we going to do about enhancements and selection of humans and babies? And broader, this is more of a social science question, a broader one. There's at least an idea that as people get more and more exposure to their DNA data and can act on it, both in selecting their partners and their children, whether there's going to be a further separation of people who have access to those technologies and have quote-unquote good genes versus those who don't and can act and can't act on them. And we call this the genotocracy, which is like the meritocracy, but only based on your genes. So that's a lot of darkness. I'm going to at least have a slide of light, which is there's just an incredible possibility. I wish I had better figures of this over all genetic conditions of very quickly and relatively cheaply and easily having a monumental reduction in the burden of genetic disorders. Just wiping out genetic disorders from the planet. That's possible in our lifetimes, and just the scale of that change in human suffering is monumental. It's something to really be optimistic about. If we just remove humans from the equation of gene editing, there's a lot of enthusiasm about editing of plants and animals, which we don't have as much ethical we don't have as much ethical issues over so far. And creating mosquitoes that don't transmit malaria, creating fruit and vegetables that do everything better than they used to. So you can really imagine, world hunger being very different, and so on. So there's a lot, again, to be optimistic about. It's just hard to know because everything's moving so quickly, and there's really no legal framework for most of this stuff. And there is this is the light. This is the not the dark side of personalized discrimination. This is restating this. The stories I was telling about dyslexia or autism, where you could sequence people's DNA as an embryo and align a set of interventions, both social and medical, that could really shape their lives in a positive way. That's a deeply interesting and potentially beneficial use of this information. So there's very much there's a lot of enthusiasm about that step. So I'm not going to spend time here. This was just in the New York Times last week, which is on the criminal justice side. We could probably talk about this all night too, which is that there was a death that was unsolved, and they only had a body part, and they used the DNA from that body part to reconstruct the face of the person. See, I mean, this is something that's happening right now. Now, that's the really light side, or seems like a great use. At the same time, New York State is allowing DNA searches of relatives of people. So, for example, like if your child is in the arrest data set and you did something illegal and your DNA is at the crime scene, they can find you because your relative, your child, is in their DNA set. So they didn't get you, they got your child, and your child is 50% you, and they can figure out that it's you or your twin. Right, so that is a recent, just this year, the State Commission on Forensic Science in New York has allowed this to happen, has allowed the use of searching of relatives, not just for the people who actually committed the crime. So I'll say thank you to the people who supported me in the organizations. And so there's both at the University of Wisconsin, we have the Institute for Research on Poverty and the Center for Demography and Ecology, which are lower left, and then other places like the Robert Wood Johnson Foundation, National Institutes of Health, and the William T. Grant Foundation. And I'll say thank you, and I'll open this back up for questions and have my contact information. Yeah, I'll just call your colors orange.
You say that all these letters are the same and they never change, but what about, I thought that there's new research that says you can change your genes. I could change my genes through what I do.
It's a great question. So one clarification is that I'm describing genetics, and there's a whole field called epigenetics, which is on top of the genome. Epigenomics is on top of the genome. And what that means is that the environment turns on, off, up, and down the function of your letters. My whole talk has been about the letters themselves. The environment, in part, shapes the way the letters work, whether they're on or off. For example, your diet, and this is what you're describing, your diet, some of your environment more generally. But that's not permanent. The epigenome is like, right now, if I sampled your saliva, you'd have an epigenetic set of markers of what's up and what's down now because of what you had to eat today in other experiences. That's a whole another interesting component that's like on top of this, but not against it. So to answer your question, you're exactly right. Your epigenome changes every second. Your genome does not. Yep. Blue.
I read somewhere when highly intelligent people mate with each other, whatever, that there's also a higher incidence of autism because they don't, so there's some unforeseen circumstances or consequences or unintended consequences on some of this matching.
That's right.
Going on.
Yeah. So I want to say that it's definitely true that that's a risk for autism. There's many other risks for autism are much greater than the combined, like the interaction of the IQs of your parents. But for a lot of mental health conditions, schizophrenia and so on, you have something similar where it's associated with high IQs. Yes. Yeah, over here.
I think kind of related to that point, though, is kind of the unintended consequences of making those decisions. Where if you can identify if this gene has a particular function or correlation, but there would be something else as well that you're not considering.
No, so a great example of that. So the question is, what about unintended consequences of the? Or I'm adding some language here, but there's the scientific term is pleiotropy, which is a single variant can do many things in your life. And the ApoE gene is a good example. So having a variant of ApoE puts you at, having the quote-unquote negative variant of ApoE puts your lifetime risk of Alzheimer's at like 90%. And there's a question of how does that survive in the human population of having such a destructive condition being tied to your genotype like that. And the answer is potentially that in the recent past it was great to have that variant. And there's evidence for that. So that's an example of pleiotropy, which is that the same variant does different things across your life, where it might increase your fertility as a 20-year-old, but then destroy your brain as an 80-year-old. That's right. Sorry, sweater gray?
What are your thoughts on GMOs, genetically modified Monsanto companies and Carlisle with what they're doing with these plants and how people are saying that you shouldn't eat them because of the genetically modified and they're going to affect you adversely?
Yeah, so the question was about genetically modified organisms or food in particular. So the one, let me see how to say this. There's almost no such thing as a non-genetically modified food. We're really at the cusp, though, of just like the scale of how we change the food. So there's been breeding programs forever that genetically modify the pool of chickens that are available and so on. So there's always been genetic modification. Now we're just at a different stage where we with surgical precision change letters around for traits. Now your question is very similar to like what are the unintended consequences of changing the food supply in this way. We won't know until it happens, right? It'll only be in hindsight where we discover that, that one nutrient or that one letter really affected the nutritional value of a tomato, and now no tomatoes have it. That's bad. On the other hand, genetic editing could potentially go backwards and edit it the other way. So it's something that might be recoverable after we make these discoveries about there being problems with the editing that happens. In the front, yeah.
Yeah, with that social groundswell against GMO, I mean, you could make a watermelon that's bug resistant, so you could save herbicides, you could save pesticides. I mean, there's a lot of things that could come out of that, but socially, non-GMO is preferred. How could, perhaps, how do you see that same social groundswell against what you were discussing, which is the human aspect? Would there be enough ethical outrage that could potentially prevent some of the stuff that you've identified as being potentially occurring?
I don't think ethical outrage will be enough. This seems like a legal matter to me. I think because there's just, in the short term, I think there's going to be so many advantages of using this information from both you personally and from society, or perceived advantages, I should say, that's going to overwhelm. This is just me, seat of the pants, having a prognostication, so I don't have any evidence for this. But quote unquote, knowing your risk of Alzheimer's and your risk of certain conditions, there's people. Millions of people seem to want to know exactly that information. The question is then, how is that going to play out in selection of things that we care about, your careers, your partners, and your children? And are people going to not do that on their own, meaning that they're just going to, as a personal matter, not eat GMO products, that kind of thing. I kind of can't imagine that to be the answer without a large-scale legal remedy, meaning it's just illegal to do X in the United States. Right now we don't have a lot of that. There's even a concern, though, with that on the scientific communities, which is that anything that we do in the U.S. that takes away for the types of science we could do about gene editing, as a single country, very powerful, but we're not the only one. China, in particular, does not have those issues and are going to run with this technology. So there's a global question of regulation as well. So that's certainly what we should have in our minds now. about how we might make the laws that really matter and for us, but realizing that we're not a global, we're not the global authority legally. Way in the back.
Yeah, regarding China, for example, for their pursuit of the perfect baby or whatever it was called, is that mandatory at this point? Like the government telling them they have to do it, or is it by choice? And if it makes its way to the U.S., is there any way that they could stay ahead of it in a legislative manner?
So legislatively, the U.S. could just say, you cannot test for X in embryos, where X is as broad as possible. That doesn't mean you can't do this as an American, go to China and do it, in a different country. But that is a possibility. So we have restrictions on what you can test for and when in a pregnancy. We could add or subtract from that legal framework. But it doesn't say you. This is so easy to do. You just have to have a finger prick on the pregnant mother, expected mother, and send that blood to China, and you get all the information you would in the United States. Yeah, in the front.
So similar to Social Security number, when we were young, you gave that out, fill it out on every form, and now our children, we guard that.
Yeah.
Is this similar to where like the young people in particular, their DNA should really be protected and guarded and not given out?
It's a great question. So the question was about young people, and you can only make this choice once, really, like whether you're going to submit or not. Because once you do it, it's kind of done. So this is not exactly your question. There's an interesting ethical question about genotyping, meaning collecting DNA from children, and what information you provide to the parents versus withhold because the child is a person who might not want anyone to know about those things. So now my understanding is that the information that's provided to the parent are of genetic conditions that occur during childhood. So for example, there are some mutations in the genome, BRCA1, BRCA2, for breast cancer. Because that expresses itself past childhood, that information is withheld from the parent so that the child can decide themselves when they are at risk for breast cancer later. So that's an example of withholding because, many becauses, but one is that when the parent learns about the child carrying something, one of those parents have that something. The way that the child gets these variants is that one of the parents or both passed it to them. So there's very interesting family dynamic questions about once one person is in this data set, the amount of information that it just kind of spreads to the family. Both vertically up the family line, grandparent had it or parent had it, and then across to siblings and so on, and then children. So it's an incredibly complicated question about who owns that information. Is it the child, the parent, everybody, nobody, this kind of thing? Red shirt? Yeah.
I just want to share, because my family, we did get genetic stuff done, and it's interesting because you do have a reflective part to see, like, where did I come from, and me being half adopted was interesting to me. Like, I never knew I was part Jewish, to hear these cool stories about, ooh, I discovered something. But then, like, the things you pass on, so I know you were saying, like, parents don't necessarily have access to certain things, but they do, like, in our case, we do have access to a variety of things, so.
But you mean you're an adult now when you're making the decision, or were you a child when you're making the decision?
I made the decision for my children and myself.
Yeah.
So as a guardian,
Yeah.
technically, you have access to those things. So there's a legal implication. I can see, someday maybe my kids are like, boy, I wish that maybe you didn't know that. But I think that's the place we went through. At least you get to decide, like, do I want to know this or not? And like yes, I want to know, or no, so I don't want to know. So like one of the things is like late onset Alzheimer's. Do I want to know if I have those genes or not? So I can click through and learn those things and share them with who I want to and not share them with who I don't want to. And then I also am able to see for the genetic stuff, like what did or didn't we pass on to our kids? So I'm just going to give an example, like I have 2 genes for lactose intolerance, according to like, ooh, I got a double set, ooh, probably lactose intolerant. I don't have the symptoms of that. My husband has 1 copy, and he has symptoms of lactose intolerance. So we can see though, like what our kids got for a matchup. But the nice thing is, if they start having symptoms, at least we're able to say, you know, this might be the cause, and you could go from there. So it has some benefits too, I guess, is my part that I'd like to share, as far as knowing a little bit about for your kids to hopefully make things a little easier for them in life, so they're not having a sore gut one day going, oh my gosh, I feel yucky all the time, because you already have a pretty good clue as far as what may be going on for them.
Yeah, absolutely. Thank you for sharing that. In the back? Yeah.
Are there any previous historical, you mentioned this revolution, any comparable historical social things that you could maybe learn from?
I'm all ears about that. I quizzed my students, my undergrads who are following, who are basically taking the course you just saw. But we were really future forward. We were because my point my point was that this is just one of the revolutions that people of whatever age you are now will like go to the next generation and say like, this was not at all what I was like 20 years ago. We didn't know nothing about this. This is a game changer, and there's not very many of these. This isn't answering your question because I don't know. I can't think of one that I think was so short in temporally, like how quickly this has happened and how we're kind of behind figuring out what to do with it. But then going forward, I figured I'd share what the class came up with are global warming, climate change is something that's this relatively short, huge implications in artificial intelligence as being something that you're going to reflect back onto the generation of your children or grandchildren, saying like that was just nothing of my experience growing up, and now it's everywhere, or now it has fundamentally changed human society. So those are the couple thoughts. But we all put this as one of those possibilities that's going to fundamentally change the way we interact with each other and the types of policies we have. Who knows? I don't know if it's going to be true, but these are just our thought exercises about filling in the list of like big things, big changes in society that are going to have really large implications. Backwards, I don't know. I'd be interested if you have or anyone in the crowd has a thought on going back 50 years or 100 years of something that was so abrupt, that was a technology that was so abrupt that had maybe these type of social implications for it in the front row.
Something simple to think about would be an RH chapter. Back when it was in the 60s, Rogan came to be where you would have children and the mother had a negative blood type, the fetus had a positive blood type, and then the child would be born jaundiced. They'd be undergoing. We remember that.
Yeah.
Then it was known testing for the ABO group, and now Rogan came to have protection against the fetus from the parental maternal antibodies, so that made healthier babies. And so simple genetics was in play way back then.
That's right. Yeah. But this it's just the scale, I think. It's not that, yeah, I totally agree. Yeah.
I was just wondering if, with this, genomic revolution, if that has gotten into any governments working with anything and looking at ways to help the poor, more creative, evolved ways, or
The help to the poor that someone would argue for is the reduction in genetic conditions in the next generation. And this is going back to gene editing now, the potential treatment of people who are currently adults who have genetic conditions. Myopia and blindness is an example where there's, I'm trying to think, it's just the last few weeks the FDA has been approving some drugs for gene therapy. One of them was for blindness that, just a very relatively non-invasive injection of the right cells, the gene cells that have been edited, provide people with sight. The question is, how much do companies get to charge for that technology? The companies believe it's like your value of eyesight your whole life. So millions of dollars is the answer to the company side. So the question of like what health insurance covers is going to go forward, because all these technologies are going to start, or I should say, all the therapies are going to start with really high price tags, right? Partly because they're targeting rare conditions that not many people have. So to recoup the money that was spent for R&D, they're going to charge a lot for it. But that's not the only answer. They're going to, blindness affects a lot of people worldwide. What are we going to do for providing access to those treatments where no individual would have enough money to pay $1 million for their eyesight their whole life? And what are we going to do for Medicaid budgets when we say yes or no to these treatments and it just explodes the whole budget for a state? It wouldn't take many treatments of this type that are so expensive that if Medicaid covered it, it would be the whole budget for that year. What are we going to do as a society for those type of therapies? Because they're ongoing. So we're just now in the first few months of the first sets of FDA-approved gene therapies that could go at scale now. So we're just weeks into that. But the number of trials that are in stage 2 or 3 are in the dozens. So going in the next 6 months, this is going to be something very much on people's plate to deal with. Who's going to pay for that? Any other questions?
Bridging the Reins. I think you all know that it was certainly worth walking through the chilly temps to listen to Fabulous presentation. As we said, we'll be doing another one of these in the spring, but certainly watch our chapter Facebook page and our website, the Wisconsin Alumni Association.